So what we do, all these biopsy specimens, I'll be talking about that, transporting and plating the samples, the pathologist perspectives. So when you have a specimen like that, which can be non-infectious, masquerade, or infectious, so that infectious can be viral, bacterial, fungal, and parasitic. So pathologic study techniques can be AC tap, iris biopsy, vitreous biopsy, or retinochoroidal biopsy, as has been told by Dr. Sarvanan. So in case of fluid, what do you do it? Intraocular fluid, which can be direct smear. If it is a little bit more, you can do the cytospin. Then culture, which is a gold standard. Polymerase chain reaction, so now should be used in infectious infectious uveitis or endophthalmitis. Immunohistochemistry and flow cytometry from the tissue sections or cell block preparation. Cytology, how do you do it, is a cytospin for concentration of the specimen, where the fluid is put in the funnel and then kept in the cytospin machine, and 1,000 revolutions per minute for 5 minutes. And you get a concentrated smear like this, which can elegantly show the cytology of the specimen. So one can do that in various uveitic entities like anterior chamber aspirate in a pseudo-hypopian, and this can show the retinoblastoma cells, or lens-induced uveitis where macrophages engulfing lens material, or retinochoroidal biopsy in case of large cell lymphoma showing necrotic background large lymphoma cells. What about culture? Culture is the gold standard, but less sensitive, takes too much time, and not practical for virus infection. How do you transport the intraocular specimens to microbiology? It has been told. What it can do, the sterile broad tube with a rubber band inside to which the needle is inserted and sent to the laboratory so that the anaerobic conditions can be maintained. And it should be sent as soon as possible in the micro lab within 30 minutes to maintain the anaerobic conditions. And this has been told, the growth of bacteria culture or fungus cultures, which you can see in the plates. In case of primary vitreoretinal lymphoma, one tissue diagnosis is mandatory. Here, the tissues are often necrotic background, so it should be transported within half an hour to 1 hour. And cytologic smears new astute cytopathologists because they can be confused with the necrotic background and few large lymphocytes, the infiltrations which will be seen because the inflammatory component is there. What we do is that also not only the cytology, but we can do the cell block preparation. The fluid is put into that one vial, and the sediment is taken and put into the cytospin. The paraffin block is prepared, and which can be processed, sectioned, stained, and viewed on histologic section. It's easier to do special stain, immunohistochemistry, or cell block slides. And here is a cell block preparation from the vitreous aspirate showing elegantly pleomorphic lymphoma cells with necrotic background. So diagnostic efficacy of cell block is a high degree of sensitivity and low pseudopositivity rate. So how do you do that? Instead of vitreous biopsy, as has been told, core vitrectomy is better. At least core vitrectomy leads to post-personal vitrectomy is better. Primary intraocular lymphoma, alcohol fixation causes poor cellular recovery. So send the specimens without any preservative within half an hour to the laboratory. And what we see, the pathologist is allowed round or oval nuclei, often segmented with the prominent nucleoli with scant basophilic cytoplasm. This is the showing that lymphoma cells with the pleomorphic large lymphocytes with the large nuclei. And you can see the cell block preparations showing the necrotic background lymphoma cells, which can be stained with the CD2 B cell marker or CD79A B cell marker. Molecular biology has come in a very revolutionized our looking into the infectious etiology in various specimens. And one get that 10⁶, 1 million copies of the DNA can be seen over here. And what you require is very small, tiny samples and primers for the specific detections, nucleotides, and enzymes. And the specimens can be taken from any part of the eye. Even if you have forgotten to take the specimens for PCR, you can take from the paraffin section of the eyeball, scrape it, and the DNA can be amplified by PCR. This is a PCR machine. The beauty of it is that the report is ready within 12 hours. And this has got inbuilt positive control and the negative control. So it detects the high degree of sensitivity and specificity. So various infectious agents can be detected by PCR. It includes a host of bacteria, fungus, or viruses. Many more are getting added every day. Every time there's one. There are several types of PCR is there. I won't go to the details of it. Single step PCR, nested PCR, multiplex PCR, quantity PCR, and last but not the least is the generation sequencing, next generation PCR with the sequencing and direct sequencing. Single step PCR is a one target, one step reaction, 20 to 40 cycle, and you get the CMV, HSV, VZV, Toxoplasma gondii, as well as the tubercular uveitis. Nested PCR, two sets of primer is used. Amplified DNA becomes the second primer, and you will get the amplified product. Advantage is that increased specificity and the increased sensitivity, but there is a risk of contamination and the time and cost. The application is very widely used in mycobacterium tuberculosis DNA estimations. An example of a nested PCR, this we thought that primary intraocular lymphoma in the vitritis was there with materials in the vitreous cavity, but histopathology showed granuloma, and we confirmed with the PCR that it's a mycobacterium tuberculosis infection with MPV 64 and IC102-Topic region positive. So multiplex PCR, a single PCR reaction can detect multiple pathogens. Each primer set generates amplification of a specific size different from the other. And the multiplex PCR is the advantage is the simultaneous detection of multiple pathogens at one time, cost and time saving, but this disadvantage is the decreased sensitivity due to the primer competition compared to the uniplex. CMV, HSV, VZV, Toxoplasma gondii can be detected. Reverse transcripted PCR, where you can use that enzyme reverse transcriptase and can be used in the influenza and bacteria through the ribosomal RNA. Real-time PCR is a further advancement. Here you get the copy number, fluorescent molecule is used to monitor the PCR reaction. An example of real-time PCR is the case of acute retinal necrosis, where you can get the viral copies, 88,740 copies, and you can get that the card, fluorescent card, which detects the, compared to the normal card and detects the viral copies. So PCR is a very useful in the vitreous sample for the viral retinitis. We have studied the 100 suspected cases of infectious uveitis. It confirmed or provided new diagnosis in 70% cases. How to send the sample for PCR? Acetab and vitreous sample used in the syringe. The movement should be arrested by sealing the syringe with abdo-tape or adhesive tape, or you can put in the vial and make it in the sterile, and you can put in the three package, the primary package, secondary package, and tertiary package to make sure that the fluid doesn't leak out. What's new is the metagenomic diff sequencing, which can identify common and rare pathogens. And last but not the least is the MYDL265 mutations, which can be done in the intraocular lymphoma from the vitreous fluid with a sensitivity of 88.9% and specificity of 91.6%. In conclusion, ocular biopsy sample is often small but very precious. Proper handling of the material is of paramount importance. You need to discuss, as told by Dr. Manisha, that with pathologists of your clinical differential diagnosis prior to the biopsy. So pathology can give importance clue to our clinical practice. This is told by William Osler, a great clinician, as your pathology, so is our practice. Thank you very much.